Hello everyone, today we're going to present about the genome-wide transcriptome analysis of human epidermal melanocytes using multiple bioinformatics programs. So, the human epidermal melanocyte is a type of cell on our skin that synthesizes photoprotective pigment, melanin, which helps provide critical protection against skin cancer, and they are located on the basal layer of the epidermis.
So, why is this research important? Skin cancer is the most common form of cancer in the United States. Skin cancer is divided into two groups, non-melanoma and melanoma categories. Melanoma is caused by transformation of pigment-producing cells known as melanocytes. Although melanoma is less common, it is a more serious type of skin cancer. Melanoma contributes to over 75% of all skin cancer deaths and accounts for around 9,000 deaths in the US annually. Therefore, better understanding of melanocyte function and human skin pigmentation is needed to discover treatment of skin cancer and pigmentation disorders.
And now we explain to you this research pipeline, which is called the tuxedo suit. So first, they use Illumina HiSeq 2000 to perform RNA-seq. Next, they use FastQC to check the quality of the raw and RNA-seq data. Next, they use Bowtie 2 to align RNA-seq reads to the reference genome. And they use Picard tools to identify the functional class of the read.
They then use the top hat to identify the splice junctions, cufflinks to assemble transcript and estimate relative abundance, and EdgeR to identify which genes are differentially expressed. The file format, say from Illumina HiSeq 2000, is .fastq, until Bowtie 2, where it is changed to .sam. Illumina HiSeq 2000 is a powerful high-throughput sequencing system which functions to perform RNA-seq. It is used to examine the quantity and sequences of RNA, such as mRNA, tRNA, or rRNA. It uses a method called sequence by synthesis, also known as SPS chemistry, both which I will elaborate more on later. The input files format is a binary-based call, also known as .bcl, and can be converted into a text-based sequencing data file format, also known as .fastq. Now onto RNA-seq. RNA sequence is a technique that can examine the quantity and sequences of RNA in a sample by analyzing the transcriptome of gene expression patterns encoded within our RNA. This provides us with a more sensitive and dynamic way to study its expression, as the results obtained contain less noise and have higher specificity. The benefits of RNA-seq is that it provides us with quantitative data at single base resolution, information on transcript size, and it is not limited to the number of known genes and transcript isoforms at the time of study. This data can also be reanalyzed as sequence databases are updated. Now onto SPS. Sequence by synthesis enables parallel sequencing of millions of fragments using a method called reversible terminator base, which detects single bases as they are incorporated into a growing DNA strand. This occurs as the first sequencing cycle begins by adding 4 labeled reversible terminators, primers, and DNA polymerase. After laser excitation, the emitted fluorescent from each cluster is captured and the first base is identified. The cycle then repeats itself with the incorporation of the 4 labeled reversible terminators, primers, and DNA polymerase. After another laser excitation, the image is captured and the identity of the second base is recorded. These sequencing cycles are repeated to determine the sequence of bases in a fragment, one base at a time, which end result is a true base-by-base sequencing that enables accurate data for a broad range of applications.
After sequencing using Illumina, the quality of the raw sequencing data was assessed using FastQC. FastQC is a tool that can be used to check the quality of raw sequencing data from high-throughput sequencers such as Illumina. Fast can be imported in the form of BAM, SAM, or any FastQC file variants. The tool provides a quick overview that tells you in which area there may be a problem. The file is exported as HTML, and it provides various information on the sequence. The first is basic statistics, which shows simple information on the input file. Next is per base sequence quality, which is a box and whisker plot that tells the quality from the beginning to the end of the sequence. It gives statistics on the per position basis of the qualities. Then we have per sequence quality score, which displays a graph showing the average quality per read. Typically, a quality score above 20 is desired, and graph should be fairly tight in the upper range of the plot. Next is per base sequence content, which reports the proportion of each 4 of the nucleotides, adenine, thymine, cytosine, and guanine. It also displays per base GC content, which is also proportions but from the GC perspective. And per sequence GC content, which shows the theoretical distribution curve determined from the GC content and actual GC content curve. It also shows per base N content, sequence length distribution, sequence duplication levels, which indicates how many reads are represented more than once, overrepresented sequences, which lists sequences that appear more than expected, and it also indicates adapters or other known sequences. And lastly, k-mer content, which is another way of looking at overrepresented sequences, but not for full sequences. Rather, it is for short nucleotide lengths. It will also display bias of a particular sequence at a certain position. In this paper, The quality scores of the reads for each library and for each position in the read were above 30, which is defined as high quality.
Bowtie2 is a program that aligns sequences reads to long reference sequence with multiple supported formats. The input files that they use are FASTQ, QSEC, and FASTA files. They are used to quantify how similar the read sequence is to the reference sequence using a score. The higher the score, the more similar it is. As well as the higher the score, the more unique it is for the mapping quality, and they output their results in a .sam format. The Bowtie2 build command builds an index from a reference genome sequence from a public genome database. They have multiple options to allow multiple input formats such as FASTA files or raw reference sequences. You are also able to name the final output file in a .bt2 format. The Bowtie2 align command maps long or short sample sequences reads against reference sequences. They support multiple types of input formats such as FASTQ, FASTA, or raw sequences files. You are also able to name the final output file in a .sam format. In order to map a human epidermal melanocyte, a Bowtie2 build command is used to generate a .pt2 file by using grch38 human chromosome melanocyte as reference sequence. Bowtie2 align command is then used to align the RNA sequence in FASTQ format generated by Illumina and reads it to the reference genome generated by the Bowtie2 build using grch38 as reference sequence. A score is then generated based on the similarity.
And now for the pickup tools. We are using one of the functions under pickup tools called collect RNA-seq matrix to obtain our RNA matrix by aligning sample RNA to functional classes of loci in the reference genome. To use these pickup tools, you need prior coding knowledge, but we've actually found a website called Galaxy, which has optimized UI to be used by anyone without any coding knowledge. And the input file is SAM file, and this tool is used for, firstly, obtain RNA matrix such as ribosomal bases, coding bases, untranslated region bases, and so on and so forth. Why do we need all these metrics? Is to enhance the quality of our sample RNA by reducing the interference from rRNA or tRNA. Because for this research, we only want mRNA, so by reducing the interference from other RNAs, we can enhance our quality of our sample RNA. And the output file is SAM file.
Moving on to the next bioinformatics tool would be the TopHat. TopHat is a tool that aligns RNA sequence reads to genome using Bowtie or Bowtie2 and map to a reference genome to identify exon-to-exon splice junctions. As seen in the diagram, TopHat can be run on free servers through Galaxy, which also provides a web-based genome or track browser for mapped reads produced from TopHat. The input file accepted for this tool would be SAM file, FASTA, and FASTQ file formats. It is to be noted that TopHat does not support mixing FASTA and FASTQ reads in the same input file, so it is advised not to run TopHat on FASTQ and FASTA files in the same run. The requirements for this tool would be Bowtie2 and Bowtie2 align or Bowtie, Bowtie2 inspect or Bowtie inspect, Bowtie2 build or Bowtie build, and SAM tools. To start the TopHat pipeline, the usage command as attached should be entered. The output files are SAM file or BED file format. The workflow of TopHat is summarized in the diagram attached. RNA sequence reads are mapped against the whole reference genome using Bowtie. TopHat identifies potential exons since many RNA sequences will contiguously align to the genome. An initial consensus of map regions is computed and assembled by MAC. Sequences flanking potential donor or acceptor splice sites within neighboring regions are joined to form a database of potential splice junctions. TopHat collects unmappable reads and constructs seed table index. For each splice junction, TopHat searches the initially unmapped reads in order to find reads that span junctions using a seed and extend strategy. The initially unmapped reads are indexed and aligned to these splice junction sequences.
And now for cufflinks. We are using cufflinks for two reasons. First is to assemble the aligned read obtained from TopHat's output in a set of transcripts and to estimate the relative abundance of our transcripts. And the input file is SAM file, and it is used for first, assemble short reads to long transcripts by, as you can see in this picture. First, identify the mutually incompatible fragments, next connects all fragments by overlapping, and lastly construct the transcript isoform. And the second step is to calculate the transcript abundance by assigning undetermined fragments to transcript isoforms. And the output of these cufflinks is also SAM file. And now I will show you the results of cufflinks from the research paper. This is the result, and they are calculated in FPKM, which is fragments per kilobase of exons per million fragments mapped. And they are able to identify transcript abundances, which are in red color, by comparing it to the blue color housekeeper gene. And FPKM equals to the level of gene expression. So when you have higher FPKM, it means that your gene is highly expressed.
As for the result of the research of transcriptome analysis of human epidermal melanocytes, a comparative transcriptomic analysis of lightly versus darkly pigmented HEMs using high-throughput sequencing technologies revealed a novel set of 15 genes and 166 transcript isoforms in which 17 isoforms are involved in signal transduction. The novel set of genes and isoforms are potentially related to differences in skin pigmentation because they are differentially expressed in lightly and darkly pigmented HEMs. The research findings suggest that the putative melanosomal transporter SLC45A2 may be a key regulator in melanogenesis, which provides a fundamental foundation for discovering pharmacological targets responsible for the treatment of melanoma or other pigmentation disorders.
Thank you for watching, and we hope you enjoyed the video. Have a nice day.